Hi everyone, I'm Rayk, the principal investigator at the Institute of Clinical Chemistry and Clinical Pharmacology at the University Hospital and the Medical Faculty of the University Bonn. With this little teaser, I'd like to draw your attention to our manuscript Deficiency for SAMHD1 activates MDA5 in a cGAS sting-dependent manner. It was just now published in the Journal of Experimental Medicine. Sensing of endogenous nucleic acids has been shown to cause systemic autoimmunity like the monogenic type 1 interferon-driven disease Aicardi-Goutier syndrome. One of the genes mutated in AGS patients is the DNA repair enzyme SAMHD1. And previous work on SAMHD1 showed that loss of the enzyme leads to interferon production via the cGAS sting pathway in response to spontaneous DNA damage. So we generated SAMHD1 knockout mice and found spontaneous DNA damage in these mice. The DNA damage had actual functional consequences because when we crossed SAMHD1 knockout mice to a tumor-prone background, loss of SAMHD1 decreased the tumor-free survival of p53 deficient mice, suggesting increased spontaneous DNA damage in the double deficient mice. Surprisingly, this did not further increase the interferon response, arguing against the strong impact of DNA damage in regulating interferon response in SAMHD1 knockout mice. So this left us with the question of how interferon is induced. To address this, we performed a series of genetic in vivo studies. First, inactivation of the essential proteins MAFS and STING in the Rayk-like receptor and cGAS sting pathways demonstrated that both pathways are simultaneously required for the interferon production in SAMHD1 knockout mice. Of the two Rayk-like receptors, only inactivation of MDA5 but not of Rayk-I abrogated the interferon response, suggesting that MDA5 drives interferon production in cells lacking SAMHD1. So what about cGAS sting signaling in this model? When comparing transcriptomes, we and others before us observed a lower transcription of ISGs, including many RNA sensors in cGAS deficient mice compared with controls. This reduced Rayk-like receptor expression compromises cytoplasmic RNA sensing in cGAS knockout cells, resulting in a higher tolerance against cytoplasmic double-stranded RNA ligands. The expression defect of Rayk-like receptors in cGAS sting deficient cells can be overcome by ectopic expression of MDA5, which leads to an increased interferon production in SAMHD1 sting deficient cells and further suggests the presence of endogenous MDA5 ligands in SAMHD1 knockout mice. In summary, we showed that in healthy cells, a cGAS sting-dependent tonic interferon signaling primes Rayk-like receptor-dependent cytoplasmic RNA sensing. This enables MDA5-dependent interferon production in SAMHD1 deficient cells that can be abrogated by inactivation of cGAS and termination of tonic interferon signaling. Our work suggests that endogenous MDA5 ligands drive the disease in Aicardi-Goutier syndrome patients lacking SAMHD1, and illustrates an important role of cGAS-dependent tonic interferon signaling in nucleic acid immunity. That's it for now. Check out our manuscript for more findings related to SAMHD1. We're happy to share our resources or answer questions that you might have. Take care.